kind of this diagram that you can download from Moodle. It's on, it's under, I think, brain or under the peripheral autonomic nervous system. But this is a handout. It's a 2-page handout, and this is the second page. The first page really is kind of walking through that big diagram that was on the previous slide, and then noting what types of receptors are found at different organs. Now this is interesting, right? We have two systems, parasympathetic and sympathetic. And we have two different types of receptors for those neurotransmitters or hormones or neurohormones. We have cholinergic receptors, and those are parasympathetic, and we have adrenergic receptors, which are sympathetic. Hopefully that makes sense. Acetylcholine and adrenergic, right? The two types of cholinergic receptors that we have are muscarinic and nicotinic. Nicotinic are by far the most numerous. They're all over the place. Acetylcholine touches nicotinic receptors all over the body. And then adrenergic, we have really 5 different types of receptors, but we're only going to look at 3 of them here. Oops, I wonder if I can get rid of that. The 3 that we're going to look at, or the 4 that we're going to look at, I'm sorry, are alpha. We have 2 alphas, alpha 1 and alpha 2, and beta 1 and beta 2. Now, the question becomes, hey, where do I find these receptors? Go look in a textbook somewhere. You cannot memorize these. They pretty much don't make a whole lot of sense. But what does make sense is that we make a split between the ones, so alpha 1 and beta 1. These are both stimulatory. And then we have alpha 2 and beta 2. These are both inhibitory. So we plus or minus on the organ, stimulate the organ or inhibit the organ. All right. So now we'll go back to this diagram here. Parasympathetic nerves, we see long preganglionic, short postganglionic. What happened to my color there? That's interesting. And we have this dotted line here. That is our ganglion. And we have, oh, you know, I think I can, maybe I can't. I'm going to enlarge this here. So this is the parasympathetic nervous system. And in the circle, I'm going to put down the receptor, and on the line I'm going to put down neurotransmitter. I'm going to put another circle right there too. In the ganglion we have nicotinic receptors, so you can download this and mark it up yourself. We have nicotinic receptors, and because it's parasympathetic, our neurotransmitter is always acetylcholine. Then at the level of the organ, for most things, we have muscarinic receptors, and we still use acetylcholine. Okay? This is the standard. Things don't change much from that. Okay? What will change is the action on the organ. Okay? But let's go over to the sympathetic side. And the sympathetic side is a little bit more complex, right? Because we have not only nervous input, we also have hormones. We have the adrenal medulla, which we'll take a look at. When we look at sympathetic nervous system, we see the same thing. We see nicotinic receptors in the ganglion and acetylcholine. Now down here at the end, we're not going to put in a receptor because we can choose one of 4 kinds. So we're going to have to think about, we're going to have to leave that sit for a minute. But our neurotransmitter is always going to be norepinephrine and epinephrine. And that's going to be all over the place. When we get to the adrenal medulla, so this is our adrenal medulla, we have particular cells in there that have nicotinic receptors, and they respond to acetylcholine, and they release epinephrine and norepinephrine, kind of in those quantities, 80 to 20. Sometimes you'll see it a little bit different, 85, 15 or whatever. Okay? That's our standard. So we'll see at the ganglion, which is in the adrenal medulla or in the sympathetic chain, nicotinic receptors and acetylcholine. Same thing over here in the ganglion, nicotinic receptors, acetylcholine. That's always. At the level of the organ, we see parasympathetic is muscarinic, and it's the neurotransmitter is still acetylcholine. At the level of, or at the sympathetic, at the level of the organ, we're going to have to figure out what receptor it has, but it always has norepinephrine and epinephrine as the neurotransmitters. Let's take a look at the heart and see what we mean here. If we're looking at the heart, and we do parasympathetic, that's easy. Nicotinic, acetylcholine. Now, parasympathetic, so we're going to leave that for a second on the myocardium in the heart. And on the sympathetic side, we know it's going to be norepinephrine and epinephrine. How do we know that? Because I just told you that, all right? And that's an always. What's different here is we have to figure out what type of receptor is here. Now, I would certainly recommend go look it up. But we do know one thing. Do I want to speed up or slow down the heart? I want to speed it up. So if I go back and take a look at my note from up here, I said the ones, alpha 1 and beta 1, both circled in red, are always stimulatory. Alpha 2 and beta 2 are always inhibitory. All right, so now go back here. What do I want? Do I want ones or twos? You better have just said, I want ones, because that's what it is, right? I want to stimulate the heart. And it just happens to be beta 1. Again, how do I know that? Because I looked it up. Maybe I can, that's terrible. I'm going to try this again. Beta 1. Okay, and we know that ones are stimulatory, right? And that's fight or flight. I'm going to speed up the heart. And acetylcholine and nicotinic, I said these are muscarinic. My mistake. Erase that from your brain. These are always muscarinic. And that's going to be inhibitory. So beta 1, I couldn't fit the beta 1 in my circle there. So beta 1, stimulatory, muscarinic, acetylcholine, inhibitory. All right. We'll take a look at another one here. Do we want to go over here? Nope, we want to go over here. We're going to drop down to here, to the bladder. All right, we'll do the same thing. We have parasympathetic, so these are both, there's muscarinic on the muscles in the bladder wall and muscarinic at the sphincter. We have acetylcholine and acetylcholine. It's always that. On the sympathetic side, epinephrine and norepinephrine. Epinephrine and norepinephrine. Always. Now the question becomes, what are the receptors? And again, you'd have to know or go look it up. Well, let's take a look at the parasympathetic side first. What do we want the bladder to do under parasympathetic control? Well, one of the things that I use to help remind me what the parasympathetic does is SLUDD, which is salivation, lacrimation, urination, digestion, and defecation. All right, so now here we are. We're at urination, right? So if I'm going to urinate, what am I going to do to the sphincter? Do I need to tighten it or relax it for urine to come out? You're right, we're going to relax it, so that's a minus. What about the muscle on the bladder wall? Do I want to contract it or relax it? Absolutely correct. We want to contract it. Okay, so this will contract the bladder wall and loosen the sphincter, and urine will come out. Okay, so what we want to do under sympathetic is the opposite. I want to have this be a minus, that's the opposite of the plus, and I want this to be a plus, which is the opposite of the minus, right? So that tells me that I'm going to have a 2 up here because 2's were inhibitory, and I'm going to have a 1 down here because 1's are stimulatory. And again, I can go back to a chart and find out that this is going to be an alpha 1 and this will be a beta 2. Okay, how do I know that? Again, I went and looked it up. But I could figure out, is it going to be a 2 or a 1? Now take a look at this. You probably have heard or have seen medications to lower your heart rate. And hopefully this makes sense. This is why we work on this, because pharmacology is based on these receptors. Okay? If I can block beta receptors, I will slow the heart down. Beta blockers, that's what slows the heart down. So I'm blocking this, I'm stopping the sympathetic stimulation. Unfortunately, one of the side effects of beta blockers is urinary incontinence. I'm going to block this beta too. That's going to keep the muscle relaxed. So if I block this one, because of autonomic tone, that takes over. So I contract the muscle on the bladder and I squeeze out urine. That's what causes urinary incontinence when using beta blockers. I could get around this because this is a 2 and that's a 1 if I used beta 1 blockers exclusively rather than just beta blockers. Probably more expensive. Okay? So here we have the gastrointestinal tract. Oops, I'm going to squeeze this down a little bit. And we have all of these receptors and neurotransmitters to work on. We'll go through parasympathetic first. Easy. It's muscarinic and acetylcholine. It's muscarinic and acetylcholine. It's muscarinic and acetylcholine. Always. Okay. Now what's the action? If I'm going to hit the gastrointestinal tract with acetylcholine on a muscarinic receptor, what's it going to do? Speed it up or slow it down? Well, which one of these D's doesn't really matter. Digestion. I'm going to want to churn my stuff, so this is going to be a positive. Okay? Because I'm digesting, I'm also going to stimulate my glands because I want to keep my stuff in the gastrointestinal tract lubed up and digesting, so I'm going to plus that too. The other D was defecation. So what's my action on the sphincter here, which is what that is. That has to be a minus, so I relax that sphincter so I can defecate. Hopefully that makes sense. We'll go through here and look at the sympathetic side. The sympathetic side is going to be the opposite, right? This is a minus here on the sphincter, so I want to make that plus. This is a plus on the wall of the gastrointestinal tract, so I want to make that a minus. And this is a plus down here, so I'm going to make this a minus. So I can go back and assign my numbers. It's a 1, right, because it's plus. This is a 2. And I should be drawing these inside those little circles. 1, 2, and then this has to also be a 2. The neurotransmitters norepinephrine and epinephrine, norepinephrine and epinephrine, norepinephrine and epinephrine. Okay, sorry it's so sloppy. Now I'm going to go back and look and see which type of receptors those are. This happens to be alpha 1, and that as an alpha 2, and that's an alpha 2. Okay. Now, so this doesn't go on forever. We have others on here. So go ahead and see if you can figure out the airway and the skin vessels and heart, lung, and skeletal muscle vessels, notice, think about this, there's only one circle and one line. There's only one circle and one line. Okay? That means it's not dually innervated, it's singly innervated. The other thing that you could also do, you should also do, I guess, is, oops, there's my, that's like the worst sweat gland I've ever drawn. There's sweat glands. Take a look at a big chart on receptor types. This one, the sweat gland, is totally different than anything else we've seen, which is why you should take a look at it. It's also singly innervated. Okay? Airway, dually innervated, you can see all those. If you have any trouble with those, get in touch with me and we'll walk through it. We'll figure it out. But I went through enough on the example side. Hopefully you can get the rest. And you have to think about, so what you have to do when you think about it is, do I want, under fight or flight, do I want blood to go to the gastrointestinal tract and skin vessels? Under fight or flight, do I want blood to go to my heart, lung, skeletal muscle? Under fight or flight, do I want my airways wider or narrower? Under fight or flight, do the sweat glands activated or not? Okay, so those are the things you have to think about when you're answering those. And then hopefully, using a chart like this and your knowledge of this chart, when you go in to look at peripheral neuropathies, you can tell the whole idea of looking at peripheral neuropathies is finding out, is it, it's autonomic. Yes, yes, granted. But what side? Is it a parasympathetic problem or is it a sympathetic problem? And using the chart here, you should be able to tell, because there are some key systems under sympathetic that do not have parasympathetic innervation. And because of that, we can use that knowledge to find out, is it sympathetic or parasympathetic? Am I overstimulated on sympathetic or am I understimulated on sympathetic? Am I overstimulated on parasympathetic or am I understimulated on parasympathetic? Those are your four choices, and use those systems to figure out what's going on. Hello. We're going to take a quick look at the sympathetic and parasympathetic nervous system. This may help you figure out some information, hopefully some information to use in solving some of the quests out there. The sympathetic and parasympathetic nervous system obviously are both part of the autonomic nervous system, or the ANS. So there are two divisions, and what we see between them, as you can see kind of in the center of this diagram, that the parasympathetic nervous system and the sympathetic nervous system both have nerves coming out and innervating organs like the eye. So we have parasympathetic and sympathetic input. We can also see the same thing with the pancreas, right? We have both parasympathetic and sympathetic input. Intestines, okay, both parasympathetic and sympathetic. And also on this diagram, you can see one of the, or some, I guess, of the main features and differences between the sympathetic and parasympathetic is the sympathetic comes out of the thoracolumbar region, and it has short preganglionic and long postganglionic fibers, and the ganglion is in the sympathetic chain right there. So we have a sympathetic chain ganglion, and neurons come out, synapse right there. So we have a preganglionic neuron coming out of the spine, and then the postganglionic neuron coming out from the ganglion. Contrast that with the parasympathetic nervous system, where we don't have a ganglion, really. We do. It's closer to or within the target organ. But here we see a long preganglionic and then a very short postganglionic fiber. And we see the same thing, maybe a little bit easier here, long preganglionic, and then we have some little ganglion there, and then a short postganglionic fiber. Okay, so there's some anatomical difference between those, and you can also see that the neurons that come out of the parasympathetic system are craniosacral. So we're coming out of the spine and really midbrain and medulla oblongata at different regions than we do with the sympathetic nervous system. Okay? So we call this idea of both systems working at the same time dual innervation. And between the two, what we'll see is autonomic tone. If I go back to this screen, we can see that almost all organs have innervation from both systems. However, these guys out on the side are only, they're away from the parasympathetic, right? These guys are only innervated by the sympathetic nervous system. Okay, and so what do we have? We have hair follicles, we have sweat glands, and we have peripheral blood vessels. Okay, that means they are only activated under fight or flight. Okay, parasympathetic does not tell your sweat glands to stop. What tells your sweat glands to stop? Shutting down the sympathetic nervous system. Okay, so hopefully that makes sense. So, and lastly then, we have, what does that mean? Autonomic tone means that if we have stuff, just like your own muscles, have tone, like if you're alive, right, your muscles have tone to them. If I'm getting information from both the sympathetic and the parasympathetic at the same time on a single organ, then we have autonomic tone and this dual innervation. Okay, what does that mean? Kind of in reality here is you can kind of think of the autonomic nervous system. It's always on, and if we had a little gauge right here, we'd see that we're kind of always pointed towards the parasympathetic. Now it can move. You know, if you start to go into fight or flight, the needle will move to the other side. When you relax, the needle moves back. But at rest, we kind of sit just kind of slightly towards the parasympathetic nervous system. So that's kind of where our body sits. And that's what autonomic tone creates for us. And then we shift back and forth between those two systems that are both always on.